Hello, everyone. Today we are going to discuss the topic of gene expression and regulation. You might wonder, since every cell in our body carries exactly the same DNA, why do some cells become neurons, others muscle cells, and still others liver cells? The answer lies in gene regulation. Even though the DNA sequence is identical, each cell selectively turns on certain genes and off others. This process is what we call gene regulation. Gene expression refers to the entire process by which information in a gene is used to synthesize RNA and subsequently proteins. Regulation determines when this process is initiated, how much product is made, and where the gene is expressed. These events are not random. Rather, they are controlled through highly precise cellular mechanisms. In other words, our body relies on the selective switching on and switching off of genes to define the identity and function of each cell type. In this lecture, we will begin with the fundamentals of transcription and translation, then examine how prokaryotes regulate gene expression through operon systems. We will then explore the diverse regulatory strategies in eukaryotic cells, such as chromatin modifications, transcription factors, and even RNA-mediated regulation. By the end, you will understand why the same stretch of DNA can give rise to the entire diversity of cell types and organisms. Pretty fascinating, isn't it? Let's get started. All right, the first thing we need to talk about is this question. How does genetic information flow and get transmitted? In other words, how do genes control the traits of living organisms? To begin with, all hereditary characteristics of an organism are encoded in the nucleotide sequence of DNA. Each segment of DNA acts like a code, instructing the cell on when, where, and what kind of protein should be produced. And why are proteins so important? Because virtually all cellular functions and structures rely on proteins. In this sense, proteins serve as the bridge that connects genotype to phenotype. This entire flow of information is summarized in a very famous concept known as the central dogma of molecular biology. It states that the information stored in DNA is first transcribed into RNA, and then RNA is translated into protein. In short, DNA, RNA, protein. This process occurs in two main stages. One, transcription. A specific segment of DNA is copied into a complementary strand of RNA, more specifically messenger RNA, mRNA. Two, translation. The mRNA is transported to the ribosome, where its sequence is decoded, amino acids are assembled one by one, and ultimately a functional protein is synthesized. Through this flow, cells transform stable genetic information into dynamic biological activity. This is precisely why understanding this mechanism is so essential. It explains how traits arise, how diseases develop, and even how biotechnology can be used to manipulate life itself. I'm sure most of you have seen examples of animals with albinism, such as white tigers, white snakes, or even pure white peacocks. These strikingly unusual appearances are often the result of a single gene malfunction. But why can such a small genetic change cause such a dramatic difference in the whole animal? The key lies in the function of the affected gene. It normally encodes a protein responsible for producing pigments. For instance, melanin is the primary substance that gives color to skin, eyes, and hair. When this gene is mutated, the resulting protein may lose its function, or it may fail to be produced at all. As a consequence, the animal's body cannot synthesize pigment properly, and its skin, hair, or even eyes appear white or pinkish. The pink color comes from underlying blood vessels showing through. This illustrates a fundamental principle. Sometimes a single gene mutation is enough to produce a highly visible change in an organism's appearance. It stands as a classic example of the close connection between genes and proteins. Now let's take a step back and ask, how did scientists first discover that genes determine traits by directing the production of proteins? This idea can be traced back to the 1940s with the pioneering work of George Beadle and Edward Tatum. Using the bread mold Neurospora crassa as their model organism, they observed that when a particular gene was defective, the mold lost the ability to synthesize certain essential compounds, such as specific amino acids. As a result, the organism could not grow. From these experiments, they proposed a groundbreaking hypothesis known as the one gene, one enzyme theory, that each gene is responsible for producing a single enzyme. Later, however, scientists realized that not all proteins are enzymes and that many proteins consist of multiple polypeptide chains. Thus, the concept was refined into the one gene, one polypeptide hypothesis, which remains foundational today. Moving into the 1950s, there was still intense debate. Are genes made of DNA or proteins? Landmark experiments by scientists such as Oswald Avery and later Alfred Hershey and Martha Chase ultimately demonstrated that DNA is the true genetic material. Finally, in 1958, Francis Crick articulated what we now know as the central dogma of molecular biology. Genetic information flows from DNA to RNA and from RNA to protein. This framework established the canonical pathway of gene expression, the transmission of information from DNA to protein. Through this process, genes shape not only our appearance and cellular functions, but also our health and susceptibility to disease. Earlier, we mentioned that genes are responsible for producing proteins, and one particularly important class of proteins is enzymes. But how was this concept discovered? Let's look at a landmark historical experiment, the 1944 study by Serb and Horowitz, which extended the work of Beadle and Tatum. They also worked with the bread mold Neurospora crassa, a remarkable organism that can synthesize all of its required amino acids if provided with only the most basic nutrients. In their experiments, Sierp and Horowitz exposed the mold to X-rays to induce mutations and then examined which mutants failed to synthesize certain amino acids. Specifically, they focused on the metabolic pathway for arginine synthesis, ornithine, citrulline, arginine. What they found was striking. Some mutants could only produce ornithine. Others could proceed as far as citrulline. Still others were blocked at the step leading to arginine. What does this tell us? It became clear that each mutation disrupted a specific step in the metabolic pathway, and that each step required a distinct enzyme. Crucially, each of these enzymes was encoded by a specific gene. From this evidence, they articulated the one gene, one enzyme hypothesis. In other words, a gene functions as the switch that controls a particular biochemical reaction. This study provided some of the earliest and clearest experimental proof for the idea that genes determine biochemical functions, laying the foundation for modern molecular biology. In the process of gene expression, transcription and translation are the two essential steps shared by all living organisms. However, there are some clear differences between how these processes occur in bacteria, prokaryotes, versus eukaryotes. Let's start with bacteria. In bacterial cells, DNA is located directly in the cytoplasm since they lack a nucleus. As soon as a gene is transcribed into mRNA, that mRNA can almost immediately be engaged by ribosomes for translation. In other words, transcription and translation occur simultaneously, a phenomenon known as coupled transcription and translation. This allows bacteria to produce proteins very quickly, perfectly suited to their fast-paced reproductive lifestyle. In contrast, things are much more complex in eukaryotes. Here, DNA is enclosed within the nucleus, and transcription takes place inside the nuclear compartment. The initial RNA product, called pre-mRNA, must undergo several processing steps before it can be used. These include the addition of a 5' cap, a 3' poly A tail, and RNA splicing to remove introns and join exons together. Only after these modifications are complete does the mature mRNA exit the nucleus and enter the cytoplasm for translation. In other words, in eukaryotic cells, transcription and translation are spatially and temporally separated, with an additional layer of post-transcriptional processing in between. While this may take more time, it also provides eukaryotes with greater opportunities to regulate gene expression, such as producing alternative splicing variants, controlling where the mRNA is expressed, and determining when translation occurs. This confers a much higher degree of flexibility. To summarize, although transcription and translation serve the same fundamental function in both systems, converting genetic information into proteins, the way they are executed is quite different. Bacteria are fast and streamlined, like a factory assembly line where production flows directly from one stage to the next. Eukaryotes, by contrast, are multi-step and highly regulated, more like a film that must be edited, cut, and subtitled before its release. These differences reflect how each type of organism optimizes the flow of genetic information into the working molecules of life. Class, we all know that proteins are made up of long chains of amino acids, and the precise order of these amino acids determines the shape and function of a protein. But here's the question. DNA contains only 4 letters, the 4 bases A, T, C, and G. How can just 4 letters encode the information for more than 20 different amino acids? It's a clever system. Instead of using a simple one-to-one relationship, one base for one amino acid, cells read the genetic code in groups of 3 bases. Each group of 3 nucleotides is called a codon, and each codon specifies one amino acid. Why groups of 3? If only one base were used, there would be just 4 combinations, A, T, C, G, far too few. If 2 bases were used, there would be 4 squared equals 16 combinations, still not enough. But with 3 bases together, we get 4p equals 64 possible codons, which is more than enough to cover the 20 amino acids used in proteins. In the genetic code table, every amino acid is represented by at least one codon. Many amino acids have 2, 3, or even 4 codons. This phenomenon is known as codon redundancy or degeneracy. One advantage of this system is that if a point mutation occurs in the DNA, the codon may still specify the same amino acid, so the protein can remain functional. There are also a few special codons. OG is the start codon, signaling the beginning of translation, and it also encodes the amino acid methionine. UAA, UAG, and UGA are stop codons. They do not code for any amino acid. Instead, they instruct the ribosome to terminate protein synthesis. In summary, although DNA has only 4 letters, by reading them 3 at a time, cells can generate 64 codons, sufficient to precisely encode all 20 amino acids. This entire system is known as the genetic code, and remarkably, it is nearly universal across all organisms, a powerful piece of evidence supporting the common origin of life. Class, we now know that the genetic code is organized into codons, groups of 3 nucleotides in DNA or RNA that specify amino acids. But did you know? The entire set of 64 codons was already deciphered by scientists back in the early 1960s. Out of these 64 codons, 61 codons specify amino acids. In other words, they instruct the ribosome which amino acid to add next during protein synthesis. The remaining 3 codons are special. These are the stop codons. They do not correspond to any amino acid. Instead, they signal to the ribosome that the protein chain is complete and translation should end. One very important feature of the genetic code is that it is redundant, but not ambiguous. What does that mean? Redundant. Several amino acids can be specified by more than one codon. For example, leucine has 6 different codons that all encode it. This redundancy provides flexibility and tolerance against certain mutations, not ambiguous. Each codon always specifies only one amino acid. A codon never stands for 2 different amino acids. This ensures accuracy and precision in protein synthesis. Finally, there is one more critical concept. Codons must be read in the correct reading frame. If the ribosome begins grouping nucleotides from the wrong position, say, starting at the second or third base instead of the first, the triplet grouping shifts, the entire amino acid sequence changes, and the resulting protein is likely non-functional. That is why the start codon, usually AEG, is so crucial. It establishes the correct reading frame for the entire translation process. This figure uses the firefly gene expressed in plants and animals as an example to illustrate that the ability of genes to function across different species provides strong evidence for evolution. Scientists have successfully inserted the firefly luciferase gene, the gene responsible for bioluminescence, into plants and even into pigs. Remarkably, under certain conditions, these organisms were able to glow as well. You might wonder, plants and animals are so different. How could they possibly use the same piece of DNA? That's an excellent question, and the answer lies at the heart of evolutionary biology. These experiments demonstrate that the fundamental structure of genes and the mechanisms of gene expression are shared across species. In other words, whether in a firefly, a corn plant, or a pig, the cells recognize the same DNA signals, know how to read the gene, transcribe it into RNA, and translate it into protein. This indicates that even though species appear vastly different, their genetic language and operating machinery come from a common ancestor. This is what we call evolutionary conservation. Therefore, beyond their practical applications in agriculture and medicine, these cross-species gene expression experiments provide compelling evidence that all life shares a common origin, or evidence for evolution. 英文课堂一篇讲稿采用篇重点版，把 Firefly gene case shared mechanism evolutionary conservation 条列整理。When we say that DNA must be converted into protein, the very first step is transcription, the process of copying the genetic information in DNA into an RNA molecule. This process can be divided into 3 stages: initiation, elongation, and termination. In the initiation stage, the transcription machine called RNA polymerase must first locate the correct starting point on the DNA. This is a specific sequence known as the promoter. Think of it as RNA polymerase asking, where exactly should I begin copying? Once it finds the promoter, it unwinds the DNA double helix, like unzipping a zipper, to expose one strand of DNA that will serve as the template for RNA synthesis. Next comes the elongation stage. Here, RNA polymerase begins moving along the DNA template strand, adding RNA nucleotides one by one and linking them together. Remember, in RNA, uracil U is used in place of thymine T to pair with adenine A. So the RNA chain grows as the polymerase advances, much like a typewriter moving forward while continuously typing out the message. In this way, the DNA information is faithfully copied into RNA. Finally, in the termination stage, the process comes to an end. When RNA polymerase reaches a termination signal on the DNA, it stops transcription, releases the newly synthesized RNA molecule, and detaches from the DNA. At this point, the transcription of that particular gene is complete. Altogether, transcription is the mechanism by which cells take a segment of DNA information and accurately convert it into RNA. This RNA will then move on to the next stage, translation, to guide the synthesis of proteins. Class, we just mentioned that transcription can be divided into 3 stages, and the very first step, initiation, is the most critical. In eukaryotic cells, transcription initiation is much more complex than in prokaryotes because it is not carried out by RNA polymerase alone. Instead, it requires the coordinated action of an entire team of proteins. First, the cell identifies a DNA sequence upstream of the gene known as the promoter. This region acts like a signpost that says, transcription starts here. One particularly important promoter element is the TATA box, located about 25 base pairs upstream of the transcription start site. The TATA box is recognized by a protein complex called a transcription factor, specifically the complex TFIID. Once TFII binds to the TATA box, it recruits additional transcription factors. Together, these factors build a platform that allows RNA polymerase II to bind securely. When RNA polymerase II, along with all the necessary transcription factors, assembles at the promoter, the structure is called the transcription initiation complex. After the initiation complex is fully assembled, RNA polymerase II unwinds the DNA double helix to expose the template strand and prepares to begin RNA synthesis. You can think of this entire initiation process like a stage rehearsal before a performance. The lighting, sound, host, and stage crew must all be in place before the main performer, RNA polymerase II, can step onto the stage and begin copying the script of life. Now let's move on to the second stage of transcription, called elongation. You can think of this stage as the moment when RNA polymerase officially begins typing. As it moves along the DNA template strand, it adds new RNA nucleotides one by one. At this point, RNA polymerase is firmly attached to the DNA. It unwinds a small section of the double helix, creating what is known as a transcription bubble, and then reads the template strand. Following the rules of base pairing, A pairs with U, T with A, C with G, and G with C. It links RNA nucleotides together into a growing RNA chain. During elongation, RNA polymerase works with remarkable efficiency. It can synthesize 20 to 50 nucleotides per second. And it's not just moving forward blindly. It manages the process as it goes. The DNA ahead of it is unwound, the DNA behind it rezips into its double helix, and the newly synthesized RNA strand emerges out the back, no longer bound to the DNA. The RNA produced is complementary to the DNA template strand and nearly identical to the coding, non-template strand, a kind of duplicate copy. You can picture RNA polymerase as an automated typewriter, scanning the original manuscript, DNA, while typing out a fresh transcript, RNA, all while cleaning up after itself, unzipping ahead, reclosing behind, and neatly spooling out the new RNA strand. In short, the elongation stage is the continuous copying of information from DNA, allowing the RNA molecule to grow longer and longer until the polymerase eventually encounters a termination signal. Once RNA polymerase has successfully synthesized an RNA molecule, it eventually needs to stop. After all, transcription cannot go on indefinitely. This brings us to the final stage of transcription, termination. In prokaryotes, there are 2 common mechanisms, intrinsic termination and row-dependent termination. Let's start with intrinsic termination. This pathway does not require any additional proteins. RNA polymerase itself will stop. How does this work? It relies on the structure of the RNA. When the polymerase transcribes a specific DNA sequence, the RNA forms a hairpin loop structure. Immediately following the hairpin is typically a stretch of uracil, U, residues. The hairpin causes RNA polymerase to slow down, and the downstream U-rich region binds weakly to the DNA template. As a result, the RNA-DNA hybrid becomes unstable, the transcription complex disassembles, RNA polymerase falls off, and the RNA transcript is released. Now let's look at row-dependent termination. Here, a key player comes into action, the Rho protein. Rho is an enzyme that can bind to RNA and move along it, essentially chasing RNA polymerase. When RNA polymerase slows down or pauses at certain sites, Rho catches up. Using its helicase activity, Rho pulls the RNA transcript away from the DNA template, thereby ending transcription. Because this mechanism requires the help of an external protein, it is referred to as Rho-dependent termination. In summary, intrinsic termination relies on RNA's own structure, the hairpin plus U-tail, to make RNA polymerase disengage. Rho-dependent termination relies on the Rho protein to actively pull off the RNA. Both mechanisms ensure that transcription ends precisely and accurately, preventing RNA polymerase from running off indefinitely. Together, they are essential quality control systems for transcription in prokaryotic cells. So how does transcription termination work in eukaryotes? With RNA polymerase II, the process is much less straightforward than in prokaryotes. After transcribing a specific sequence known as the polyadenylation signal, for example, AAUAA, RNA polymerase II does not stop immediately. Instead, it continues to synthesize a short stretch of RNA downstream. At this point, a set of specialized processing enzymes cut the RNA at the proper site and then add a poly A tail to the new transcript. Meanwhile, RNA polymerase II itself disengages from the DNA through a series of more complex mechanisms, gradually releasing rather than stopping abruptly. As you can see, the termination process in eukaryotic cells is considerably more complicated. Unlike prokaryotes, there is no simple hairpin structure or row protein to instantly bring transcription to an end. In simple terms, prokaryotic termination is like a fast stenographer. Once the end symbol appears, transcription stops immediately via a hairpin loop or row protein. Eukaryotic termination is more like a formal meeting closure. There are official steps to follow, including RNA cleavage and the addition of a poly A tail before everything is properly wrapped up. These differences reflect the greater structural complexity and regulatory capacity of eukaryotic cells. By now, we know that in eukaryotic cells, the RNA produced right after transcription is only a primary transcript, pre-mRNA. It cannot be used immediately for protein synthesis because it contains not only useful information, but also many extra segments. These unnecessary stretches are called introns, while the coding segments that actually contribute to proteins are called exons. The next step for the cell is to carry out an essential processing step known as RNA splicing. In simple terms, RNA splicing is like editing a video. You cut out the irrelevant clips, the introns, and then stitch together the meaningful footage, the exons, to produce a polished final version, the mature mRNA. This editing task is performed by a remarkable molecular machine called the spliceosome. The spliceosome is a complex made of both RNA and proteins, capable of precisely recognizing the boundaries between introns and exons, removing the introns, and ligating the exons together. What's even more fascinating is that splicing is not always done in a single way. The same pre-mRNA can be spliced in different patterns to generate different mRNAs. This is called alternative splicing. It's like having the same raw video footage, but editing it into multiple versions, a comedy, a documentary, or a drama. For the cell, this means one gene can give rise to multiple protein products, greatly expanding efficiency and functional diversity. To summarize, the purpose of RNA splicing is to remove introns and join exons, producing a mature RNA that can actually be translated into protein. Alternative splicing takes this one step further, allowing cells to generate many different proteins from a relatively small number of genes. Quite impressive, isn't it? The process of copying DNA turns out to be much more sophisticated than it first appears. Earlier, we said that pre-mRNA contains both introns and exons. So here's the key question. How are introns removed, and who does the cutting? The answer is a remarkable molecular machine known as the spliceosome. You can think of the spliceosome as the cell's molecular level editing crew, responsible for recognizing the splice sites on pre-mRNA, precisely removing introns, and joining exons together. How does the spliceosome work? First, it identifies three critical signals within each intron: the 5' splice site at the beginning, the 3' splice site at the end, and a special adenine nucleotide known as the branch point. The spliceosome itself is a large complex made up of small nuclear ribonucleoproteins, SNRNPs, pronounced SNRPs. These SNRNPs use base pairing interactions between their RNA components and the pre-mRNA to lock onto the correct positions, like a GPS guiding the editing machinery with pinpoint accuracy. Once assembled, the spliceosome first cuts the RNA at the 5' splice site. Then the released end folds back and covalently links to the branch point A, forming a looped structure called a lariat. Next, the spliceosome cuts at the 3' splice site, removes the entire intron, and finally ligates the two exons together. The result is a continuous, mature mRNA that can be translated into protein. This process is both highly precise and wonderfully complex, carried out by the collaboration of RNA and proteins. Even more fascinating, sometimes the cell chooses to remove only certain introns or combine exons in different ways, a process known as alternative splicing. This allows a single gene to produce multiple protein products with different functions. To summarize, the spliceosome acts like a molecular pair of scissors and a tailor. It accurately removes the unnecessary introns, stitches together the useful exons, and produces a mature mRNA ready to participate in protein synthesis. We already know that exons are the portions of a gene that are retained and ultimately translated into protein. But here's an interesting question. Are exons just randomly pieced together? Or is there a structural relationship between exons and proteins? The answer is yes, there is, and the correspondence is often quite precise. Many studies have shown that in numerous genes, each exon frequently corresponds to a specific functional domain of the protein. These domains are like functional modules of the protein, responsible for binding other molecules, transmitting signals, or carrying out specific chemical reactions. For example, if a protein has 3 different domains, its mRNA is often organized into 3 major exons, with each exon encoding one domain. This modular design gives tremendous flexibility. During evolution or through splicing, changes or rearrangements in exons can create new proteins with different functions, without the need to design an entirely new protein from scratch. This is also why alternative splicing is so important. It allows cells to produce different combinations of protein domains from the same stretch of DNA, much like rearranging Lego blocks to build different functional models. To summarize, many exons map directly onto protein domains, and this modular architecture makes biological design more flexible and efficient, while also providing evolution with a powerful opportunity for recombination and innovation. Next time you look at the arrangement of exons in a gene, think of them as puzzle pieces that may assemble into the functional features of a protein. We have just learned about transcription, the process of copying genetic information from DNA into mRNA. Now the next question is, how does this mRNA get turned into protein? This is where another essential step comes in, translation. In simple terms, translation is the process by which the cell converts the code written in mRNA into a chain of amino acids that forms a protein. This takes place in the cytoplasm and is carried out by a molecular machine called the ribosome. You can think of translation as treating mRNA like an instruction manual. Each group of 3 nucleotides, called a codon, is read as a signal for one specific amino acid. Helping in this process is a remarkable adapter molecule called tRNA, transfer RNA. Each tRNA works like a delivery vehicle. On one end, it carries a specific amino acid cargo. On the other end, it has an anticodon, a sequence complementary to the codon on the mRNA. When the correct tRNA pairs with the matching codon on the mRNA, the ribosome links its amino acid onto the growing chain, like stringing beads one by one. Gradually, these amino acids form a polypeptide chain, which folds into a functional protein. So the basic idea of translation is converting the language of RNA into the language of proteins. It's a bit like translating a recipe, taking an English recipe, translating it into Chinese, and then cooking the dish accordingly. In this analogy, mRNA is the recipe, tRNA is the delivery worker, and the ribosome is the chef's assembly machine. This process is highly precise yet beautifully systematic, and it is one of the fundamental steps that enables cells to function properly. Now let's meet one of the key players in the protein factory, tRNA, transfer RNA. During translation, tRNA acts as the delivery worker, carrying amino acids to the ribosome. But it doesn't just deliver cargo, it can also read the instructions written on the mRNA. How does it manage to do both? The secret lies in its unique structure. A single tRNA molecule is only about 70 to 90 nucleotides long, yet it folds into a characteristic cloverleaf structure. You can imagine it like origami. After a precise series of folds, the molecule takes on a 3-leaf shape. Within this structure are several important regions to remember. At the bottom, there is the anticodon loop, which contains the anticodon, a sequence that pairs specifically with a codon on the mRNA. At the 3 end, the tail, a specific amino acid is attached. In other words, one end of tRNA is designed to read the genetic code, while the other end is designed to carry the matching cargo. It's a brilliantly efficient design. Even more impressive, each tRNA is matched to only one specific amino acid. This matching is guaranteed by a special class of enzymes called aminoacyl tRNA synthetases. These enzymes charge each tRNA with its correct amino acid, ensuring that errors are almost non-existent. It's like a barcode scanner in a warehouse. You'll never deliver apples to a site that ordered bananas. To summarize, tRNA is the bridge of translation. One end decodes the RNA, the other delivers amino acids, allowing the ribosome to assemble proteins precisely according to the instructions in mRNA. Without tRNA, the entire protein factory would grind to a halt. Such a small molecule, yet so elegantly designed and indispensable, truly remarkable. Last time, we talked about how tRNA serves as the delivery worker, carrying amino acids to the ribosome. But here's the question. How does each tRNA know which amino acid to carry? And who attaches the correct amino acid to the right tRNA? The answer is today's star player, aminoacyl tRNA synthetases. You can think of these enzymes as the master matchmakers of translation. Their job is to assign the correct amino acid to the correct tRNA with extraordinary precision. Here's how they work. Each synthetase recognizes a specific type of tRNA based on its anticodon and other structural features. Once identified, the enzyme chemically links the correct amino acid to the tRNA. There are 20 different aminoacyl tRNA synthetases, one for each amino acid. Each synthetase is highly specific. It only recognizes its own amino acid and the corresponding tRNA, never mismatching. This step is called tRNA charging, which is like loading the right cargo onto each delivery worker before sending them off to the ribosome. This accuracy is absolutely critical. Why? Because the ribosome itself does not check whether the attached amino acid is correct. It only reads the anticodon on the tRNA. That means the entire accuracy of protein synthesis depends on the aminoacyl tRNA synthetases doing their job correctly. In short, aminoacyl tRNA synthetases serve as the quality control checkpoint at the very beginning of translation. If they make a mistake, the entire protein will be built incorrectly. So even though these enzymes may work behind the scenes, their importance is just as great as the ribosome itself. We already know that translation is the process of converting the genetic code in mRNA into a protein. But how does the whole process actually begin? This brings us to today's focus, the initiation stage of translation. You can think of this stage like the preparations before a play begins. The opening line must be found, the main character must take the correct position, and the stage must be set before the performance can proceed smoothly. In eukaryotic cells, this process begins with a fully processed mature mRNA, that is, an mRNA that already carries a 5-cap and a poly-A tail. The small ribosomal subunit, 40S, associates with a special molecule called the initiator tRNA. This tRNA carries a specific amino acid, methionine met, and its anticodon is designed to pair precisely with the start codon AG on the mRNA. With the help of various initiation factors, the small subunit scans along the mRNA from the 5' end until it encounters the first AG codon. At this point, the alignment is complete. The large ribosomal subunit, 60S, then joins, forming the complete 80S ribosome. The initiator tRNA is positioned in the P site, peptidyl site, and translation is officially ready to begin. Next, let's meet one of the cell's most important master engineers, the ribosome. This tiny structure is the factory responsible for protein synthesis. In simple terms, its job is to read the code on mRNA and join amino acids together one by one, ultimately building a complete protein. So how does it accomplish this? Let's start with its structure. A ribosome is made up of two subunits, a large subunit and a small subunit. In eukaryotic cells, the small subunit is 40S and the large subunit is 60S, which together form the 80S ribosome. Note the S here stands for sedimentation coefficient, not addition. These two subunits normally exist separately, but when translation begins, they assemble on the mRNA and start working. When in action, the ribosome functions like a 3-station assembly line with 3 main binding sites. The A site, aminoacyl site, is where incoming tRNAs carrying amino acids enter. The P site, peptidyl site, holds the growing polypeptide chain. The E site, exit site, is where the empty tRNA molecules leave. The process works like this. When a tRNA loaded with an amino acid enters the A site, the ribosome helps attach its amino acid to the polypeptide chain held in the P site. Meanwhile, the old tRNA is shifted to the E site and then released. This cycle repeats, adding amino acids one after another until the protein is completed. What's even more fascinating is the nature of the ribosome itself. It is a ribonucleoprotein, meaning it is composed of both RNA and proteins. But here's the surprising part. The ribosome's catalytic activity, the actual chemistry of peptide bond formation, is carried out not by protein, but by RNA. This means the ribosome is essentially a ribozyme. Incredible, isn't it? This also provides strong evidence for the idea that the earliest forms of life may have originated in an RNA world. We've already learned how translation begins. Once the very first amino acid, methionine, is placed in position on the ribosome, the real assembly work of building a protein officially begins. This process is called the elongation cycle, meaning that amino acids are added one after another, making the polypeptide chain grow longer and longer. You can think of it like a conveyor belt assembly line. Each round of the cycle goes through 3 key steps: entry, bond formation, and shifting. 1. Codon recognition. The ribosome reads the next 3 nucleotides on the mRNA, the next codon. The correct tRNA, carrying the matching anticodon and its amino acid, enters the ribosome's A site, aminoacyl site. If the codon-anticodon pairing is correct, the tRNA is locked in place, ready to deliver its cargo. 2. Peptide bond formation. The tRNA sitting in the P site peptidyl site is already holding the growing polypeptide chain. The ribosome now catalyzes the transfer of this chain onto the amino acid carried by the tRNA in the A site. In other words, the new amino acid is linked to the chain, and the entire polypeptide is handed over to the newcomer. 3. Translocation. The ribosome shifts forward along the mRNA by one codon. This movement repositions the tRNAs. The tRNA that was in the A site, now carrying the growing polypeptide, moves into the P site. The empty tRNA that was in the P site is shifted into the E site, exit site, and is released. Meanwhile, the next codon is exposed in the A site, ready for the next tRNA. And then the cycle repeats. tRNA enters, amino acid is linked, ribosome shifts, old tRNA exits. Each round makes the polypeptide longer, and this process continues until the ribosome eventually encounters a stop codon. We've now followed translation all the way from initiation through elongation, and finally we arrive at the last stop in the protein factory, the termination stage. As the ribosome moves along the mRNA, adding amino acids one by one, it continues until it encounters one of the special stop codons, UAA, UAG, or UGA. What makes these codons unique is that they do not correspond to any amino acid. In other words, when the ribosome reads one of these signals, it knows, time to stop. So what happens at this point? Unlike normal codons, stop codons have no matching tRNA. Instead, a protein called a release factor enters the ribosome's A site. This release factor mimics the shape of a tRNA but carries no amino acid. Its job is to trigger a hydrolysis reaction, which cleaves the completed polypeptide chain from the tRNA. At that moment, the protein is officially released and finished. Afterward, the ribosome disassembles. The large and small subunits separate, the mRNA is released, and the translation machinery resets, ready for the next round of protein synthesis. You can think of it like finishing a pearl necklace. Once all the pearls are strung, you tie off the thread, cut it, and put away your tools, ready to start again whenever needed. Let's take a moment to imagine how proteins are handled inside the cell. When ribosomes begin synthesizing proteins, some of those proteins are destined for the endoplasmic reticulum, ER, where they will undergo further folding and modifications. But here's the question: how does the cell know where these proteins are to go? The answer lies in the signal sequence of the protein. This signal sequence is usually a short stretch of amino acids located at the N-terminus of the newly synthesized protein. It contains specific features that allow the entire ribosome to be recognized by a protein complex known as the signal recognition particle. When SRP binds to the signal sequence, it temporarily halts protein synthesis. Then, SRP escorts the ribosome and the nascent protein to the membrane of the endoplasmic reticulum. Once there, SRP interacts with the SRP receptor, positioning the ribosome at a channel in the ER membrane called the translocon. The translocon acts like a gateway, allowing growing protein to be threaded directly into the lumen of the ER. Inside the ER, the protein can undergo important processing steps such as folding and post-translational modifications, including glycosylation. Once the protein is properly directed into the ER, SRP is released and translation resumes. To summarize, the key to protein targeting to the ER is the signal sequence at the protein's beginning. Through SRP recognition and guidance, the ribosome is directed to the ER membrane and aligned with the translocon, ensuring the protein is properly delivered into the ER for subsequent processing. You can think of this entire process as a highly efficient postal delivery system, making sure every package protein is delivered to the correct address on time. First, recall that bacteria are prokaryotes, meaning they lack a nucleus, and protein synthesis takes place directly in the cytoplasm. When a single mRNA molecule is translated by multiple ribosomes simultaneously, the ribosomes line up along the same mRNA strand, forming a structure known as a polyribosome, or polysome. When observed under a transmission electron microscope, TEM, these polyribosomes appear as characteristic bead-like chains. Multiple ribosomes look like tiny particles arranged one after another along the mRNA, resembling a string of pearls. This arrangement enables bacteria to synthesize many protein molecules at the same time, greatly improving efficiency. In TM images, polyribosomes can be recognized as dense clusters of particles in the cytoplasm, in contrast to single ribosomes scattered throughout. These structures are not only the active workstations of protein synthesis, but also clear evidence that bacterial cells are highly active in protein production. In summary, a polyribosome is simply the image of many ribosomes working together on a single mRNA strand. And thanks to TEM, we can directly visualize their necklace-like arrangement, a key to the rapid and efficient protein synthesis in bacteria. In prokaryotes, such as bacteria, the processes of transcription and translation are tightly linked. a phenomenon known as coupling. This is very different from what we see in eukaryotic cells, where transcription takes place in the nucleus and translation occurs in the cytoplasm, keeping the two processes separated in both space and time. In bacteria, however, there is no nucleus. As soon as an mRNA molecule begins to be synthesized by RNA polymerase, ribosomes can immediately attach to the emerging 5' end of the mRNA and start translating it into protein. This simultaneous mechanism greatly accelerates protein production and enables bacteria to respond rapidly to environmental changes. You can think of it like an assembly line. Raw material, mRNA, is being produced at the front, and at the same time, it is immediately processed into a finished product, protein, at the back, with no wasted waiting time. In summary, bacterial transcription and translation occur simultaneously and in a coupled fashion. As soon as RNA is produced, protein synthesis begins. This coupling is one of the major reasons why bacteria can grow and reproduce so quickly, adapting efficiently to changing environments. In eukaryotic cells, transcription and translation are two separate yet orderly processes. First, transcription occurs inside the nucleus. Here, RNA polymerase reads the DNA gene sequence and synthesizes a pre-mRNA transcript. This pre-mRNA is not yet ready for use. It must undergo several processing steps, including splicing, removing introns and joining exons, addition of a 5' and addition of a poly A tail before it becomes a mature mRNA. Next, the mature mRNA is transported out of the nucleus and into the cytoplasm. In the cytoplasm, ribosomes bind to the 5' end of the mRNA and begin translation, converting the nucleotide sequence into an amino acid sequence, ultimately producing a protein. Translation proceeds in 3 stages: initiation, elongation, and termination, resulting in the synthesis of a complete polypeptide chain. Throughout this process, there is a clear spatial separation in eukaryotic cells. Transcription occurs in the nucleus, while translation occurs in the cytoplasm. This separation provides additional opportunities for fine-tuned regulation of gene expression. Furthermore, the RNA modifications and splicing steps add yet another layer of control over how proteins are produced. In summary, although transcription and translation in eukaryotes are sequential, they are distinct in both space and time. Through multiple processing and transport steps, the cell ensures that proteins are synthesized accurately and at the right moment. Sickle cell anemia is a genetic disease caused by a point mutation in the gene that encodes hemoglobin. A point mutation refers to a change in a single nucleotide within the DNA sequence. In this condition, the mutation alters the codon for the 6th amino acid of the hemoglobin protein. Instead of coding for glutamic acid, a negatively charged polar amino acid, the mutated sequence codes for valine, a nonpolar hydrophobic amino acid. This seemingly small change has major consequences for hemoglobin's structure and function. Because valine is hydrophobic, hemoglobin molecules tend to stick together, forming long fibers. These fibers distort the shape of red blood cells from their normal round flexible disc shape into rigid crescent-shaped sickle cells. The sickled cells are less flexible, prone to blocking blood vessels, and have a shorter lifespan. This leads to symptoms such as anemia, reduced oxygen delivery to tissues, and vascular complications. In essence, all of the clinical problems associated with sickle cell anemia stem from this single point mutation in DNA, a striking example of how a small detail can determine destiny. To summarize, sickle cell anemia results from a critical point mutation in the hemoglobin gene. This single substitution changes hemoglobin's properties, deforms red blood cells, and triggers a cascade of pathological effects. Let's first understand what a nucleotide pair substitution is. This occurs when one pair of bases in the DNA sequence is replaced by another. For example, when an AT pair is substituted with a GC pair. Such a change alters the corresponding nucleotide sequence in the mRNA, since mRNA is transcribed directly from DNA. However, there is a special type of substitution called a silent mutation. In this case, although the mRNA sequence is changed, the amino acid coded for remains the same. How is that possible? The reason is that the genetic code is redundant. That is, a single amino acid can be specified by multiple codons. Therefore, sometimes a nucleotide change still results in the same amino acid being inserted into the protein. In most cases, silent mutations do not affect the function of the protein. They are silent, in the sense that they do not alter the amino acid sequence of the protein. That said, they can still influence aspects such as mRNA stability or translation efficiency, but they usually do not directly change the protein's structure. To summarize, a silent nucleotide pair substitution is a base pair replacement in DNA that alters the mRNA sequence, but, thanks to the redundancy of the genetic code, does not change the amino acid sequence of the resulting protein. This makes it a relatively minor type of mutation. As we mentioned earlier, a nucleotide pair substitution occurs when one base pair in the DNA sequence is replaced by another. This substitution leads to a corresponding change in the mRNA sequence and may ultimately alter the amino acid sequence of the protein. One particularly important type of substitution is the missense mutation. In a missense mutation, the change in the DNA sequence alters a codon in the mRNA so that it specifies a different amino acid. This type of mutation replaces one amino acid in the protein with another, which can impact the protein's structure and function. Sometimes the change is relatively minor, with little to no effect on the protein. However, in other cases, the change can cause the protein to lose its function or even gain an abnormal function, potentially leading to disease. For example, imagine a codon in DNA that normally codes for glutamic acid. If a single base substitution changes that codon to one that codes for valine, this results in a missense mutation. Such a substitution is the basis of sickle cell anemia, where the altered hemoglobin protein takes on an abnormal structure. To summarize, a missense nucleotide pair substitution is a base pair change in DNA that alters an mRNA codon so that it codes for a different amino acid. This, in turn, changes the protein sequence and can have significant effects on protein function. When we talk about DNA mutations, one type of small-scale change is called a nucleotide pair substitution, where one base pair in the DNA is replaced by another. A nonsense mutation is a special case of this, where the substitution turns a codon that normally specifies an amino acid into a stop codon. What does this mean in practice? During translation, the ribosome will encounter this premature stop signal and halt protein synthesis too early. As a result, the protein chain is cut short before it is fully built. The outcome is usually a truncated protein, incomplete and often non-functional. You can think of it like a machine that has only half its parts assembled. It simply cannot operate correctly. For example, suppose a DNA codon originally reads CHA, which encodes glutamine. If a mutation changes it to TAG, then in the mRNA it becomes UAG, which is a stop codon. When the ribosome reaches this UAG, translation stops and the resulting protein is much shorter than normal. To summarize, a nonsense nucleotide pair substitution is a mutation that converts a codon into a stop signal. This causes premature termination of translation, leading to truncated proteins that typically have severe functional defects. It is one of the most disruptive types of small-scale mutations. We know that mRNA codons are read in groups of 3. If this 333 reading frame is disrupted, then everything downstream is shifted and the meaning of the entire sequence changes. This is known as a frame shift mutation. Such mutations usually occur when one or two nucleotides are inserted or deleted, which alters how all the codons are grouped from that point onward. Now, if this kind of frame shift happens very early in the sequence, and the new shifted codons immediately produce a stop codon, then translation halts almost at once. This situation is called a frame shift causing immediate nonsense. Here's a simple analogy. Imagine you are reading an English sentence where every 3 letters form a word. Suddenly, at the beginning, an extra letter is inserted. Now, the spacing between words is completely scrambled, and after only a few words, you run into a giant stop sign. The sentence ends abruptly halfway through, and the message can no longer be understood. The same thing happens with proteins. Translation terminates far too early, producing a very short, incomplete polypeptide that almost always lacks any function. To summarize, this type of mutation is among the most severe because it not only shifts the reading frame of codons, but also immediately triggers premature termination, resulting in a truncated, non-functional protein. Let's first review what a frame shift mutation is. Remember, mRNA codons are read in groups of 3 nucleotides. If nucleotides are inserted or deleted in numbers not divisible by 3, for example, adding 1 or removing 2, the entire reading frame shifts downstream. It's like reading an English sentence where every 3 letters make a word. Suddenly, a letter is added or lost, and from that point onward, all the words are split incorrectly. One of the most common outcomes of a frame shift is called extensive missense. This means that from the mutation site onward, nearly all the codons are mistranslated, producing the wrong amino acids. This can disrupt the protein's original structure, often causing loss of function or even producing toxic misfolded proteins. What makes this even worse is that the ribosome may not encounter a stop codon immediately. Instead, it continues translating the scrambled message, generating a long but completely faulty protein. Unlike a frame shift causing immediate nonsense, where translation halts prematurely, here translation keeps going but produces a disastrously incorrect polypeptide. To summarize, a frame shift mutation causing extensive missense shifts the reading frame so that every downstream codon is altered. The resulting protein sequence is thoroughly corrupted, leading to severe damage to protein structure and function, and often causing significant cellular dysfunction. When we study DNA mutations, one type is called a small-scale deletion mutation, meaning that a short stretch of DNA sequence is removed. Now, what happens if exactly 3 nucleotides, 1 codon, are deleted? This kind of mutation is known as a 3-nucleotide pair deletion. Since a codon consists of 3 bases, the removal of exactly 1 codon does not disrupt the reading frame, so it is not considered a frame shift mutation. What's the consequence then? The result is that the protein is missing 1 amino acid from its sequence. The severity of the effect depends on which amino acid is deleted and how critical that position is for the protein's structure and function. A classic example is found in cystic fibrosis, one of the most common mutations being delta F508. In this case, the amino acid phenylalanine at position 508 is deleted. Even though it's just a single amino acid missing, the protein misfolds, fails to function properly, and this defect leads to the disease. In summary, a 3-nucleotide pair deletion does not cause a frame shift, but because 1 amino acid is missing, it can weaken, impair, or even completely abolish protein function, depending on where the deletion occurs. Let's talk about how E. coli cleverly regulates the synthesis of the amino acid tryptophan, TRP. Tryptophan is essential for the cell, and when it is scarce, E. coli must synthesize it on its own. But when there is plenty of tryptophan available in the environment, the cell avoids wasting energy by shutting down its internal production. The key to this regulation is the TRP operon, a classic example of a repressible operon. The TRP operon is a stretch of DNA that contains several structural genes. These genes encode the enzymes necessary for tryptophan biosynthesis. When tryptophan levels are low, the TRP repressor protein is inactive and cannot bind to the operator sequence. As a result, RNA polymerase can attach to the promoter and transcribe the operon's genes. This produces mRNA, which is translated into the enzymes needed to synthesize tryptophan. Analogy: it's like a production line that needs to stay active. The supervisor, the repressor, is not interfering, so the workers, RNA polymerase, can freely keep the line running. When tryptophan levels are high, tryptophan itself acts as a core pressor. It binds to the repressor protein, activating it. This repressor tryptophan complex can then attach to the operator sequence, physically blocking RNA polymerase and halting transcription of the operon. Analogy: the supervisor, repressor, now holds a tryptophan report as authorization and goes to the assembly line to announce, "We have enough inventory. Stop production." Thus, the TRP operon operates through negative feedback regulation. When the product, tryptophan, accumulates, it inhibits its own synthesis. This is a smart energy-saving strategy that allows E. coli to adapt efficiently to environmental changes. In detail, the TRP operon contains 5 structural genes, TRPE, TRPD, TRPC, TRPB, and TRPA, which together encode the enzymes for tryptophan biosynthesis. The operon is normally on, but under certain conditions, when tryptophan is abundant, it can be turned off. Summary: the TRP operon is a classic repressible operon. When tryptophan is lacking, operon is on, enzymes are produced. When tryptophan is abundant, operon is off, synthesis halts. This flexible system ensures that E. coli can adjust its metabolism to the environment and conserve energy. Let's imagine E. coli as a thrifty factory. It decides whether or not to produce the enzymes for breaking down lactose, depending on whether lactose is present in the environment. The control center of this process is the lac operon. The lac operon is a classic example of an inducible operon, meaning that it is normally off, but it can be turned on in the presence of lactose. The operon contains 3 main structural genes, LAC-Z, LAC-Y, and LAC-A, which encode the enzymes required for lactose metabolism, such as beta-galactosidase. Upstream lies the operator sequence. When a repressor protein binds to the operator, it prevents RNA polymerase from transcribing the operon. When lactose is absent, operon off. In the absence of lactose, there is no reason for E. coli to waste energy producing lactose-digesting enzymes. At this point, the lac repressor actively binds to the operator, blocking RNA polymerase. As a result, transcription cannot occur and no enzymes are produced. Analogy: without raw material, lactose, the factory boss, repressor, locks up the machinery, operon. Workers, RNA polymerase, are not allowed to start production. When lactose is present, operon ON, when lactose enters the cell, some of it is converted into allolactose, which acts as the inducer. Allolactose binds to the repressor protein, causing it to change shape and release from the operator. With the repressor out of the way, RNA polymerase can now bind to the promoter and transcribe the lac operon, producing mRNA and synthesizing the enzymes for lactose metabolism. Analogy: When the factory discovers lactose in the warehouse, the boss, repressor, steps aside, allowing the workers, RNA polymerase, to enter and start producing the necessary tools, enzymes, to process lactose. Bonus regulation, the glucose effect. E. coli actually prefers glucose as its primary energy source. If both glucose and lactose are available, the cell will use up glucose first before activating the lac operon. This is regulated through the CAMC-CAP system, catabolite activator protein, which fine-tunes the operon's activity. We can explore this mechanism in more detail later. Summary: lactose absent, repressor blocks the operator, operon off, no enzyme production, lactose present, repressor inactivated by inducer, operon on, enzymes produced. The LASC operon is a textbook example of an inducible negative control system, ensuring that bacteria only switch on production when necessary, saving energy and demonstrating one of the most elegant regulatory strategies in molecular biology. Earlier, we talked about how the lac operon is an inducible operon. It's normally turned off and only activated when lactose is present to lift the repression. But that's not the whole story. E. coli actually prefers glucose as its primary energy source. If both glucose and lactose are present in the environment, the cell will use up glucose first, and only after glucose is depleted will lactose metabolism be activated. This phenomenon is known as the glucose effect, or catabolite repression. So how does the lac operon know whether glucose is still available? That's where the key player comes in. CRP, CAMP receptor protein, also called CAP, catabolite activator protein. How does CRP help activate the lac operon? When glucose levels are low, the concentration of CAMP in the cell rises. CAMP molecules bind to CRP, activating it. The active CRP then binds to a specific site just upstream of the lac operon, where it helps RNA polymerase attach more efficiently to the promoter, dramatically boosting transcription. In other words, CRP acts like a helper or booster for RNA polymerase, making transcription much easier to start. When lactose is absent, operon off, no matter what. If there is no lactose, the lac repressor stays bound to the operator, blocking transcription. In this case, it doesn't matter whether CRP is active or not. Without lactose, the operon stays off. When lactose is present, but glucose is also present, operon weakly on. Here, lactose inactivates the repressor, allowing RNA polymerase to bind. However, because glucose levels are high, CAMP levels are low, and CRP is inactive. Without CRP's assistance, transcription is weak. The operon is technically on, but enzyme production is limited. When lactose is present and glucose is absent, operon strongly on. This is the optimal scenario. Lactose inactivates the repressor, while glucose depletion raises CAMP levels, activating CRP. Active CRP binds upstream of the promoter, recruiting RNA polymerase efficiently. The lac operon is fully switched on, producing large amounts of enzymes to metabolize lactose. Summary: lactose absent, repressor bound, operon off. Lactose present plus glucose present, weak on, low CAMP, CRP inactive. Lactose present plus glucose absent, strong on, high CAMP, CRP active. The lac operon is therefore controlled by a dual system. Lactose serves as the inducer to lift repression, while glucose levels, via the CAMP-CRP system, fine-tune the efficiency of transcription. We often think of gene expression as simply DNA, RNA, protein. But in eukaryotic cells, the process is actually divided into multiple stages, and each stage can serve as a critical point of regulation. It's like a production line. From design to shipping, every step can be finely controlled. Stage 1. Chromatin structure and DNA accessibility. At the very beginning, whether a gene can be transcribed depends on whether it is exposed. If DNA is tightly packed into chromatin, such as heterochromatin, the transcription machinery cannot access it, and the gene remains off. Through histone modifications like acetylation or demethylation, cells can open up certain regions, making them accessible for transcription. This represents one of the earliest regulatory checkpoints. Stage 2. Transcriptional regulation. This is the primary control point of gene expression. Cells use transcription factors, enhancers, and silencers to determine which genes should be turned on, how strongly they should be expressed, and for how long. This stage is like deciding whether to turn on the factory machines in the first place. Stage 3. RNA processing. Before mRNA leaves the nucleus, it undergoes splicing, addition of a 5-cap, and addition of a poly-A tail. Particularly, alternative splicing allows a single gene to produce different protein variants. This stage provides significant flexibility in regulation. Stage 4. mRNA export and localization. Once processed, mRNA must be exported from the nucleus to the cytoplasm for translation. Some genes also control where within the cell the mRNA is translated, an especially important feature for cells with polarity, such as neurons. Stage 5. Translational control. Even after reaching the cytoplasm, whether an mRNA is immediately translated into protein is another regulatory step. For example, cells can use miRNAs or regulatory proteins to temporarily block translation of certain mRNAs. Stage 6, post-translational modification and protein stability. Finally, once proteins are made, their activity and lifespan can still be regulated. Modifications such as phosphorylation, ubiquitination, or methylation can alter protein activity, stability, or mark them for degradation. This is like the final quality control and packaging before a product leaves the factory. Summary: gene expression in eukaryotes can be regulated at many checkpoints. Chromatin accessibility: can the gene be opened? Transcriptional control: will the gene be transcribed? RNA processing: which version of the mRNA will be produced? RNA export and localization: where will it be translated? Translational control: will it be translated immediately? Protein modification and degradation: how active is it and how long will it last? These checkpoints give cells enormous flexibility, allowing gene expression to be finely tuned according to time, location, external environment, and developmental stage. When we talk about chromatin structure, we know that DNA is wrapped around proteins called histones to form nucleosomes, like thread wound around a spool. The ends of these histone proteins, especially H3 and H4, stick out from the nucleosome. These are called histone tails, and they are not just decorative. These histone tails act as important control levers for chromatin regulation. The tails can undergo various chemical modifications, such as acetylation, methylation, and phosphorylation. Among these, the most classic and most frequently tested is histone acetylation. So what happens during acetylation? This modification usually occurs on the lysine residues of histone tails. Normally, lysine carries a positive charge, while DNA's phosphate backbone is negatively charged. Their attraction keeps the DNA tightly wrapped around histones. But when lysine is acetylated, its positive charge is neutralized. This weakens the interaction between histones and DNA, making the chromatin looser. Once chromatin loosens, RNA polymerase and transcription factors can access the DNA more easily, leading to increased transcription. That's why histone acetylation is strongly associated with gene activation. Conversely, deacetylation makes chromatin tighter, reducing gene expression. Summary: histone tails are extensions from histones that can be chemically modified. Histone acetylation occurs on lysine, neutralizes positive charges, and reduces histone DNA binding. Looser chromatin transcription machinery can enter, gene expression increases. Deacetylation tightens chromatin, gene expression decreases. Let's imagine what a typical eukaryotic gene looks like. Unlike bacterial genes, eukaryotic genes are much more complex, with multiple structural elements that determine how they are transcribed, processed, and regulated. At the very beginning is the promoter, the site where RNA polymerase and transcription factors bind. This region controls when and where the gene is turned on. Nearby or even far away are regulatory sequences such as enhancers and silencers, which can increase or decrease gene expression from a distance. Within the gene itself, there are two kinds of sequences: exons and introns. Exons are the coding regions that remain in the mature mRNA and ultimately specify the amino acid sequence of proteins. Introns are non-coding segments that are transcribed but later removed during RNA splicing. Once transcription begins, RNA polymerase produces a primary transcript, pre-mRNA, that contains both exons and introns. This pre-mRNA then undergoes extensive processing. Introns are spliced out, a 5' is added to the beginning, and a poly-A tail is added to the 3' end. These modifications create a mature mRNA, which is exported from the nucleus and ready to be translated into protein. Summary: a eukaryotic gene is a complex unit consisting of promoter and regulatory sequences, enhancers, silencers, control transcription, exons, coding sequences retained in mRNA, introns, non-coding sequences removed by splicing. The primary transcript pre-mRNA must be processed, splicing, 5-capping, and poly-A addition before it becomes a mature mRNA. This multi-step system allows eukaryotic cells to precisely regulate gene expression and protein production. In eukaryotic cells, transcriptional regulation of genes is highly complex. Control is not limited to sequences right next to the promoter, but also involves distal regulatory elements called enhancers. Enhancers can be located thousands or even tens of thousands of base pairs away from the gene they regulate, yet they can greatly increase transcription efficiency. Enhancers themselves do not directly initiate transcription. Instead, they serve as recruitment platforms for specific proteins called transcriptional activators. These activators recognize DNA sequences within the enhancer and bind to them. Once bound, the activators work by inducing DNA looping, which brings the enhancer into close physical proximity with the gene's promoter. This spatial closeness allows the activators to interact with the transcription machinery, RNA polymerase, and its cofactors, stabilizing the binding of RNA polymerase II to the promoter and facilitating transcription initiation. In simple terms, enhancers and activators act as remote commanders. They don't operate the transcription machinery directly, but through looping and recruitment, they help the machinery work more efficiently. This mechanism provides cells with powerful and flexible gene regulation, allowing precise control of expression according to cell type, developmental stage, or external signals. Promoter and the TATA box. Within the promoter region, a key DNA element is the TATA box, a short AT-rich sequence. This is the crucial docking site where RNA polymerase II and general transcription factors assemble to begin transcription. However, the promoter alone is often not enough to drive high levels of transcription. That's where enhancers and activators come into play. Mediator complex, the bridge. A central player in this system is the mediator complex, a large multi-protein assembly that acts as a bridge, linking activators at the enhancer to RNA polymerase II and the basal transcription factors at the promoter. Through mediator, the activation signal from the enhancer is relayed directly to the transcription machinery, promoting efficient assembly and activation of the transcriptional complex. Analogy, a remote conference system. The TATA box is like the control console in the meeting room. RNA polymerase II is the key speaker. Enhancers and activators are the remote commanders. The mediator complex is the communication system that transmits the commander's signals to the speaker. The outcome, RNA polymerase II is fully activated and begins transcription from the promoter, synthesizing mRNA and completing the process of gene transcription. Summary, TATA box, key promoter element recruiting RNA polymerase II. Enhancer, distal DNA regulatory sequence that binds activators. Activator, proteins that bind enhancers and deliver activation signals. Mediator complex, the bridge linking activators to RNA polymerase II. RNA polymerase II executes transcription, producing mRNA. During interphase, the resting period between cell divisions, chromosomes are no longer tightly condensed into visible rod-like structures. Instead, they exist in a more relaxed state within the nucleus. Importantly, they are not randomly scattered, but folded and arranged in a highly organized three-dimensional structure known as chromosome topology. One key feature of this organization is the formation of chromosome territories. In simple terms, each chromosome occupies a specific region of the nucleus rather than tangling randomly with others. This spatial partitioning is crucial for proper gene regulation and DNA repair. On a finer scale, chromatin forms numerous loop structures. These loops bring distant regulatory elements, such as enhancers, into close contact with their target genes, enabling precise control of gene expression. The formation and maintenance of these loops depend on specialized proteins, such as CTCF and the cohesin complex. In this model, the topology of chromosomes is dynamic. When certain genes become active, local chromatin regions can open up, granting access to transcription factors and RNA polymerase. Conversely, inactive regions may become more compact. Summary. In the interphase nucleus, chromosomes are not distributed randomly. Instead, they are organized into chromosome territories. Each chromosome occupies its own distinct region. Chromatin loops bring distant enhancers and promoters together for precise regulation. Protein scaffolds, CTCF, cohesin, maintain loop stability and higher order structure. Together, these features create a complex, ordered 3D network that supports both efficient gene regulation and genome stability. Troponin T is a protein that plays a crucial role in muscle contraction. Its gene can undergo alternative RNA splicing, producing multiple mRNA variants that lead to slightly different protein isoforms. This allows a single gene to fine-tune the structure and function of its protein products depending on the muscle type, e.g. cardiac versus skeletal muscle or the developmental stage. Alternative splicing means that after a gene is transcribed into a primary RNA transcript, pre-mRNA introns are removed, but different combinations of exons can be selectively included or skipped. For troponin T, different exon combinations are used in different muscles or developmental contexts, generating isoforms that adjust the specific contractile properties of the muscle. This strategy is highly flexible. It enables muscle cells to produce a wide variety of proteins without needing multiple separate genes. This not only saves genomic space, but also allows protein expression to be precisely adapted to physiological needs. Summary. The troponin T gene uses alternative RNA splicing as a post-transcriptional regulatory mechanism. By combining exons in different ways, it produces multiple protein isoforms, each tailored to the functional requirements of different muscle tissues or developmental stages. MicroRNAs, miRNAs, are short, non-coding RNA molecules, typically about 20 to 24 nucleotides long. Inside cells, they act as post-transcriptional regulators of gene expression, fine-tuning protein production by binding to their target mRNAs. miRNAs regulate gene expression in two main ways. One, translational repression, preventing ribosomes from efficiently reading the target mRNA, thereby blocking protein synthesis. Two, mRNA degradation, promoting the breakdown of target mRNAs, directly reducing the pool of transcripts available for translation. How does this work? miRNAs associate with a group of proteins to form the RNA-induced silencing complex, RISC. Through sequence complementarity, the miRNA guides RISC to its specific mRNA target. Once bound, RISC either represses translation or initiates degradation of the mRNA, ultimately lowering protein output. This mode of regulation does not alter the DNA sequence, nor does it prevent transcription itself. Instead, it directly influences the post-transcriptional stage, making it a rapid and flexible mechanism for controlling gene expression. miRNAs play critical roles in diverse processes such as development, differentiation, proliferation, apoptosis, and are also implicated in diseases, including cancer. They are thus central players in the fine control of gene expression in eukaryotic cells. During cell division, chromosomes must undergo extensive condensation to ensure they can be accurately segregated into daughter cells. At the center of this process lies a crucial region called the centromere, the site where spindle fibers attach, making it essential for chromosome separation. Within the centromere, chromatin becomes even more densely compacted than in other chromosome regions. This is due to the presence of specialized histone variants, such as CNPA, along with other associated proteins. These proteins replace canonical histones, stabilizing chromatin and enabling a more compact structure. Such highly condensed chromatin not only protects DNA from damage, but also provides a robust platform for spindle attachment, ensuring that sister chromatids are pulled apart equally during mitosis and preventing genomic instability. In short, the condensation of centromeric chromatin acts as the structural foundation of cell division. It is the handshake point that guarantees chromosomes are safely and accurately distributed. siRNA and centromere regulation. Recent studies reveal that small interfering RNAs, siRNAs, together with associated proteins, play a critical role in regulating chromatin condensation at the centromere. The siRNA protein complex recognizes and binds specific DNA sequences in the centromeric region. It then recruits a set of histone-modifying enzymes, most notably histone methyltransferases, which catalyze modifications on histone tails, especially H3K9 trimethylation, H3K9Me3. This modification serves as a docking signal for HP1, heterochromatin protein 1. HP1 binding promotes tighter packing of nucleosomes, leading to the formation of highly condensed chromatin. Summary: centromeric chromatin condensation is essential for chromosome stability during cell division. siRNA protein complexes localize specifically to centromere DNA. These complexes recruit histone-modifying enzymes, e.g. methyltransferases. Histone modifications, H3K9Me3, attract HP1 proteins. The result is highly condensed chromatin, which ensures strong spindle attachment and accurate chromosome segregation. During early embryonic development, cells have not yet undergone clear differentiation, but they are already receiving developmental information that guides them toward distinct fates. This information comes from several major sources. 1. Maternal effect genes and cytoplasmic determinants. Even before fertilization, the mother has already preloaded the egg cell with specific mRNAs and proteins. These factors, known as cytoplasmic determinants, are unevenly distributed within the cytoplasm of the egg. After fertilization, as the zygote divides, different daughter cells inherit different sets of these molecules, influencing their future developmental pathways. For example, in frog eggs, the animal pole is enriched with proteins such as noggin, which inhibits BMP signaling and promotes neural tissue development. In contrast, the vegetal pole contains factors such as VG1, which helps induce mesodermal structures and endoderm differentiation. In this way, cells inherit distinct molecular instructions depending on their location in the egg. 2. Intercellular signaling and morphogen gradients. As embryonic cells proliferate and arrange themselves into spatial patterns, they communicate through signaling molecules such as morphogens. These molecules often form concentration gradients across the embryo. Depending on how much morphogen a given cell perceives, it activates different gene expression programs, leading to distinct developmental outcomes. 3. Positional information. Beyond diffusible signals, cells also sense their relative position within the embryo through direct contact and local signaling. This positional context further refines their developmental decisions. 4. Gene regulatory networks. Finally, all of these signals are integrated into gene regulatory networks, GRNs. Through the coordinated action of transcription factors and regulatory elements, GRNs precisely switch genes on and off, executing the developmental plan with remarkable accuracy. Summary: early embryonic cell fate is guided by multiple sources of developmental information. Cytoplasmic determinants, maternal RNAs and proteins preloaded in the egg, e.g. noggin at the animal pole, VG1 at the vegetal pole, giving cells inherited instructions. Intercellular signaling, molecules such as morphogens establish gradients that cells interpret based on position. Positional context, direct cell-to-cell interactions refine developmental choices. Gene regulatory networks, integrate all signals to ensure precise gene expression. Together, these mechanisms transform a single fertilized egg into a complex, multicellular organism with highly specialized tissues and organs. In the early embryo, there exists a population of embryonic precursor cells whose fates are not yet determined. These cells are pluripotent in the sense that they could potentially become neurons, skin cells, muscle cells, and so on. Once they receive specific signals, however, a subset of them will commit to the muscle cell lineage. This process is called determination. At the heart of this decision is a transcription factor known as MyoD, often referred to as a master regulatory gene of muscle fate. Once a cell switches on MyoD expression, it is essentially locked into a muscle developmental pathway, regardless of its original potential. Remarkably, if MyoD is artificially introduced into a skin cell, that cell can also be reprogrammed toward muscle fate, highlighting the powerful regulatory role of this gene. Following determination, these specified cells become myoblasts, immature muscle precursor cells. Myoblasts begin to express a range of muscle-specific genes, including those encoding proteins such as actin and myosin, which are essential for muscle structure and function. This stage represents cell differentiation, the transition from an undifferentiated precursor to a cell with specialized features. Finally, multiple myoblasts fuse together to form long, multinucleated skeletal muscle fibers. These fibers are the mature muscle cells capable of contraction, allowing them to participate in movement and posture control. Summary: embryonic precursor cells, initially uncommitted in fate. Determination, triggered by expression of MyoD, committing cells to the muscle lineage. Myoblasts, immature muscle cells expressing muscle-specific proteins, e.g. actin, myosin. Differentiation, myoblasts specialize and fuse, mature skeletal muscle fibers, multinucleated contractile cells with functional capacity. In cell and molecular biology, one question often arises: why do we study something as small as the fruit fly, Drosophila melanogaster, to understand human genes or cellular mechanisms? The answer is that the fruit fly is an extraordinarily powerful model organism with profound contributions to modern biology. First, its genome is much smaller than that of humans, yet over 70% of human disease-related genes have homologs in Drosophila. This means that we can model human diseases such as neurodegeneration, cancer, and developmental disorders using fruit flies. Second, fruit flies have a well-defined and rapid life cycle. From egg to adult takes only about 10 days, and a single female can lay hundreds of eggs. This allows us to quickly observe genetic variation and perform large-scale experiments within short time frames. Most importantly, Drosophila is one of the birthplaces of genetics. In the early 1900s, Thomas Hunt Morgan used fruit flies to demonstrate that genes are located on chromosomes, opening the door to modern genetics. Later, fundamental discoveries such as gene regulatory networks, cell polarity, and signaling pathways, Notch, Hedgehog, WN, etc., were first uncovered in Drosophila before being extended to mammals and humans. Additionally, the genetic toolkit of Drosophila is exceptionally advanced. Systems such as Gal4 UAS allow precise control of where and when genes are expressed or silenced. Other tools include RNA interference, CRISPR, and live imaging, making fruit flies a versatile and mature system for experimental biology. Development of the fruit fly. Let's now take a look at the developmental stages of Drosophila. 1. Egg stage. A fertilized egg undergoes rapid nuclear divisions without cytoplasmic division, resulting in a syncytial blastoderm, many nuclei sharing a common cytoplasm. Later, the nuclei migrate to the periphery of the egg and membranes form around them, creating the cellular blastoderm. 2. Segmentation. One of the most critical steps in embryogenesis is segmentation. The embryo becomes divided into repeated units that later give rise to structures such as the head, thorax, and abdomen. This process is controlled by a cascade of genes, including maternal effect genes, gap genes, pair rule genes, and segment polarity genes, a classic model for studying gene regulation during development. 3. Larval stage. After segmentation, the embryo hatches into a larva. The larva is worm-like and progresses through 3 instar stages, each involving molting. During this time, the larva feeds intensively, storing energy for the upcoming metamorphosis. 4. Pupal stage. The larva then enters the pupal stage, ceasing feeding and remaining immobile. Inside the pupa, the larval body undergoes dramatic remodeling. Old tissues are broken down, while new adult structures such as wings, legs, and compound eyes are constructed. 5. Adult stage. Finally, the adult fly emerges from the pupa with a fully developed body. At this stage, it can fly, mate, and lay eggs, beginning the next life cycle. Summary. The fruit fly is a model organism of immense importance because its genome shares strong conservation with human disease genes. It has a short, prolific life cycle for rapid experimentation. It provided the foundation for modern genetics and developmental biology. Its genetic toolkit, Gal4 UAS, RNAi, CRISPR, etc., is among the most powerful in biology. Its developmental stages, egg, segmentation, larva, pupa, adult, make Drosophila a superb system to study gene regulation, embryogenesis, and disease modeling. Let's look at a classic example from Drosophila development, how the embryo decides which end will become the head and which end will become the tail. Here, the bicoid gene, BCD, plays a central role. Bicoid is what we call a maternal effect gene. This means that the mRNA of the gene is not produced by the embryo itself, but rather preloaded into the egg by the mother fly before fertilization. Inside the egg, bicoid mRNA is not evenly distributed. Instead, it is tightly anchored at the anterior end. After fertilization, this mRNA is translated into bicoid protein, which diffuses through the embryo, forming a concentration gradient. High at the anterior, low at the posterior. You can think of this gradient as a kind of molecular map that tells embryonic cells where they're located along the head-to-tail axis. Now, what does bicoid protein actually do? Bicoid functions as a transcription factor, turning on or off other genes that are necessary for anterior development. For example, bicoid activates the hunchback gene, which in turn regulates downstream processes that specify head and thoracic structures. So what happens if a fly embryo lacks the bicoid gene? The results are dramatic. The embryo completely fails to form head structures, and instead it develops two posterior ends, in other words, a two-tail fly. This striking phenotype demonstrates just how crucial bicoid is for establishing the anterior region of the body plan. Bicoid is the textbook example of a morphogen. In fact, it was one of the first morphogens ever discovered and remains the most classic case. So, what exactly do we mean by morphogen? A morphogen is defined as a molecule that forms a concentration gradient in the embryo, and cells read this gradient to decide which genes to express and ultimately what fate to adopt. Importantly, a morphogen does not simply act as an on-off switch. Rather, different concentrations trigger different gene expression programs. Now, does bicoid fit this definition? Absolutely. In the Drosophila embryo, bicoid mRNA is deposited by the mother at the anterior tip of the egg. After fertilization, this mRNA is translated into bicoid protein, which diffuses toward the posterior, creating a gradient, high at the front, progressively lower toward the back. This gradient acts like a positional map for the embryo. Cells exposed to high concentrations of bicoid will activate genes required for head formation. Cells sensing intermediate levels will give rise to structures like the mouth parts and thorax. And cells in low bicoid regions develop more posterior structures. And the experimental evidence is striking. If scientists artificially inject bicoid mRNA into the posterior end of the embryo, an ectopic head forms right there. This is the hallmark of a morphogen. Position and fate are dictated by concentration within the gradient. First, we need to know what oncogenes are. Proto-oncogenes are normal cellular genes that play essential roles in regulating cell growth, division, and differentiation. Under normal conditions, they are tightly controlled and ensure that cells only proliferate when appropriate. The problem arises when these proto-oncogenes become overactive due to genetic alterations. In such cases, they are converted into oncogenes, which drive uncontrolled cell growth and may eventually lead to tumor formation. So what kinds of genetic changes can turn a proto-oncogene into an oncogene? The three most common mechanisms are 1. Gene translocation or chromosomal rearrangement. Here, the proto-oncogene is moved to a new location in the genome, often placed next to a highly active promoter or enhancer, leading to excessive expression. A classic example is the BCR-ABL fusion gene found in chronic myelogenous leukemia, CML. 2. Gene amplification. In this case, multiple copies of the same proto-oncogene are generated, resulting in an abnormal accumulation of its protein product. For instance, HER2 amplification in breast cancer leads to excessive growth signaling. 3. Point mutations. Even a single nucleotide change can alter the protein's activity, either making it hyperactive or resistant to normal regulation. A well-known case is the RAS gene, where mutations keep RAS in a permanently on state, continuously sending division signals. The outcome of all these alterations is the same, a loss of growth control. The signaling pathways that normally act like carefully regulated traffic lights now behave like a car with broken brakes. Cells keep dividing without restraint, setting the stage for cancer development. Summary: proto-oncogenes equals normal genes regulating cell growth. Oncogenes equals mutated or overactive versions driving uncontrolled cell proliferation. Key mechanisms: gene translocation, placed under strong promoter, overexpression. Gene amplification, multiple copies. Excessive product, point mutation, hyperactive or deregulated protein. In a healthy cell, RAS functions as a small GTPase. Under normal conditions, when the cell receives a growth factor signal, such as epidermal growth factor, EGF, RAS becomes activated. Once activated, RAS binds to GTP, switching into its on state, RAS-G-GTP. In this active form, RAS triggers a downstream signaling cascade, most notably the MAPK pathway, RAF-MEK-ERK. This cascade ultimately activates transcription factors that promote the expression of genes involved in cell cycle progression, such as cyclin D. Importantly, this signaling is reversible. After completing its job, normal RAS hydrolyzes GTP to GDP, returning to its inactive state, RAS-GDP. In this way, RAS functions like a tightly controlled on-off switch, turned on only when needed and turned off once the signal has been delivered. The problem arises when mutations occur in the RAS gene, most commonly at codons 12, 13, or 61. These mutations impair the intrinsic GTPase activity of RAS, meaning that it can no longer hydrolyze GTP efficiently. As a result, RAS remains permanently in the active RAS-GTP state. This is equivalent to a switch stuck in the on position. Even in the absence of external growth factor signals, mutant RAS continuously drives the MAPK pathway, persistently sending the message, divide. This uncontrolled signaling pushes cells into the cell cycle, leading to excessive proliferation, the first step toward tumor formation. In short, normal RAS equal controlled gatekeeper, switching on and off in response to signals. Mutant RAS equal defective faucet, constantly leaking signals, driving unchecked cell growth and oncogenesis. When cells are exposed to ultraviolet UV radiation, DNA damage inevitably occurs. To prevent these errors from being passed on to daughter cells, the cell must immediately activate a cell cycle checkpoint, pausing division until the damage can be repaired. In normal cells, this response is largely mediated by the p53 tumor suppressor protein. Upon sensing UV-induced DNA lesions, the DNA damage response, DDR pathway, activates p53. Activated p53 then induces the expression of p21, a potent cyclin-dependent kinase CDK inhibitor. p21 binds to and suppresses cyclin CDK complexes, effectively halting the cell cycle at either the G1 phase or the G2 phase. This pause provides a crucial time window for DNA repair mechanisms to restore genome integrity. Once repair is complete, the cell cycle resumes in a controlled manner. In contrast, in mutant cells, particularly those harboring p53 mutations as commonly seen in cancer, this pathway fails. Without functional p53, the cell cannot effectively sense DNA damage, and p21 is not adequately induced. As a result, cyclin CDK complexes remain active, and the cell continues to divide, despite carrying damaged DNA. This unchecked proliferation accelerates the accumulation of mutations, fueling genomic instability and increasing the risk of cancer progression. Summary: normal cells with functional p53, UV damage, DDR activates p53, p53 induces p21, p21 inhibits cyclin CDK, cell cycle arrest, DNA repair before division. Mutant cells, p53 deficient, UV damage, p53 pathway inactive, no p21 induction, cyclin CDK unchecked, cell continues dividing with damaged DNA, mutation accumulation, and tumorigenesis. One fundamental concept in cancer biology is that cancer does not arise suddenly. Instead, it develops progressively through the accumulation of multiple genetic mutations, a framework known as the multi-step model of cancer development. A classic case study is colorectal cancer, where researchers have mapped out the typical genetic events leading from a normal epithelial cell to a fully malignant carcinoma. Step 1, APC mutation, tumor suppressor gene loss. The APC gene normally suppresses the WNT signaling pathway, preventing uncontrolled proliferation. When APC is mutated and loses its function, WNT signaling becomes hyperactive. As a result, epithelial cells begin to proliferate abnormally, giving rise to a small benign adenoma, polyp. Step 2, RAS activation, oncogene mutation. A subsequent mutation in the RAS gene, commonly KRAS, locks RAS in its always-on state. This drives persistent mitogenic signaling, accelerating cell division. The benign adenoma enlarges and progresses into an intermediate adenoma with dysplastic growth. Step 3, SMAD4 or TGF-beta pathway mutations. Under normal conditions, TGF-beta signaling functions as a growth inhibitory pathway. Loss of function mutations in SMAD4 or other TGF-beta pathway components eliminate this inhibitory break. The lesion progresses into a late adenoma with invasive potential beginning to emerge. Step 4, P53 mutation, loss of the guardian of the genome. P53 is the final safeguard responsible for halting the cell cycle in response to DNA damage, orchestrating repair, or triggering apoptosis. When P53 is mutated, cells bypass apoptosis, ignore DNA damage, and proliferate unchecked. At this stage, the tumor transitions into a malignant carcinoma capable of invading surrounding tissue and eventually metastasizing. Summary: APC mutation, abnormal proliferation, early adenoma; KRAS activation, persistent growth signaling, intermediate adenoma; SMAD4 TGF pathway loss, no growth inhibition, advanced adenoma; P53 mutation, loss of DNA damage checkpoint, and apoptosis, carcinogenic cancer. When studying breast cancer, 3 molecular markers are critically important for determining both tumor biology and therapeutic strategies: ERA, estrogen receptor alpha, PR, progesterone receptor, and HER2, human epidermal growth receptor 2. 1. Hormone receptor status, ERA and PR. ER mice and PR mice, hormone receptor negative breast cancer. These tumors lack estrogen and progesterone receptors on their surface. As a result, they do not respond to endocrine therapies such as tamoxifen or aromatase inhibitors. Clinically, these cancers tend to be more aggressive and are associated with a poorer prognosis. ER plus and or PR plus, hormone receptor positive breast cancer. These are the most common subtypes. Because they rely on estrogen and or progesterone-driven signaling for growth, they are highly responsive to endocrine therapy. Prognosis is generally more favorable. 2. HER2 status, HER2 positive breast cancer. These tumors overexpress the HER2 receptor, which amplifies proliferative signaling and drives rapid tumor growth. While HER2 positivity correlates with aggressive behavior, effective targeted therapy exists. The monoclonal antibody trastuzumab, Herceptin, specifically binds to HER2 receptors, blocking downstream proliferative signaling and recruiting immune effector mechanisms to destroy cancer cells. In clinical practice, trastuzumab dramatically improves survival outcomes for HER2 positive patients and is part of standard of care treatment. HER2 negative breast cancer. These tumors do not overexpress HER2 and thus do not respond to HER2 targeted therapies like trastuzumab. Treatment decisions are based on hormone receptor status, endocrine therapy if ER/PR positive, or chemotherapy. 3. Triple negative breast cancer, TNBC, defined as ER, PR, and HER2 This subtype is the most aggressive with limited treatment options. Chemotherapy remains the mainstay of therapy, and research into immunotherapy and novel targeted approaches is ongoing. Summary: ER mice, PR mice, hormone receptor negative, endocrine therapy ineffective, aggressive biology. HER2 plus, highly proliferative but treatable with HER2 targeted therapy, e.g. trastuzumab. HER2 mice, no HER2 targeted options, therapy depends on ER/PR status. Triple negative, ERs, PR mice, HER2 mice, most aggressive, currently reliant on chemotherapy. This framework illustrates how molecular profiling directly dictates treatment decisions. Trastuzumab Herceptin serves as a classic example of precision oncology. It is highly effective in HER2 positive disease, but completely ineffective in HER2 negative tumors, making biomarker testing essential prior to therapy selection.